So let me start by thanking the organizers for this opportunity to present my work. I'd like to talk to you about modeling somatic hypermutation and then using models for hypermutation and VDJ recombination to do better at identifying B-cell clonal families. This is work I did and continue to do with my PhD advisors, Alexandra Walczak and Thierry Mora here at ENS. And so in general, the goal of our work is to build probabilistic models that can describe the diversity of clonal types that we see in B-cell repertoires, particularly as measured or as evidenced by repertoire sequencing. And so the cornerstone of this diversity is the process of VDJ recombination, where these three genes are joined together in a stochastic fashion to give rise to a large pool of very diverse sequences. In particular, there are combination junctions that find themselves in the complementarity determining region, CDR3 for short, are to large extent responsible for the specificity of a given receptor or antibody. But as we know, that's not the whole story, and B cells further diversify in the process of somatic hypermutation, where they further alter their genotype by point mutations mostly. And so what we'd like to do is to provide a probabilistic description of this process in the same way that my colleagues and my supervisors previously characterized the process of VDJ recombination, building a model which, among other things, allows to compute the probability of generating any given receptor. We'd like to extend this to somatic hypermutation in a model that would aim to characterize the likelihood of any given mutation pathway. So to fix ideas, we can perhaps think of what Catherine just told us about, which is the problem of eliciting a broad response to HIV. And as we heard, it's typically that breadth correlates with the level of mutations. And so, of course, generically, the more mutations a sequence harbors, the more any particular mutation pathway will be unlikely. But if we condition on the same number of mutations, the heterogeneity of this process causes some of them to be much more likely than others. And to quantify or to explain this variation is exactly what we set out to do here. But the profile that you see here, which is the frequency of substitutions along amino acid positions in the immunoglobulin coding gene, is not just an effect of the inherent biases of the mutation machinery, but also shaped by subsequent selection and genetic drift. And to model the mutation properties themselves, we need to disentangle the two. And to do that, we actually use the same approach that was used to model VDJ recombination, which is to look not at the productive sequences in RNA sequencing experiment, but rather at out-of-frame sequences. So products of the recombination that were actually unsuccessful and in which the CDR3 region, its length is not a multiple of 3. And so by definition, these are passenger sequences that are free of selection in the sense that whatever happens in the sequence, whatever mutations they would harbor, they don't determine the B-cell fate. And so you can see here in the CDR3 length distribution, we have a log scale, and so we have much less of the out-of-frame transcripts than productive ones. But given that we now have access to this great data sets of unprecedented depth of sequencing, this is actually enough, as I will show you, to build such models. So the data I use here for the models is the data from the Briny lab published 2 years ago. Secondly, and importantly, to get rid of phylogenetic biases and to count each mutation once, we need to reconstruct clonal families to which are present in the repertoire data. And to do that, we use standard techniques. And once the partition into lineages is done, then we reconstruct the phylogenies again using standard phylogenetic methods. What's important is that we use the templates as an outgroup that instructs that roots are 3, and in particular allows us to identify which of the mutations are shared by all sequences. And this is important because we will discard these mutations in our analysis, as some of them may be mutations and some of them may be somatic mutation, but again, as we just heard, this can also be germline mutations that we don't have in our databases, or just simple alignment errors. So this will be discarded, and we will then have all mutations on the tree, which allows us to count each and single one of them once. And the model that we built decomposes the mutation rate into context and position dependence, which is to say that we allow for mutation rate to be modulated at any given position by first the context, which is already an established knowledge that the mutation rate depends on context. And here we choose that the mutation rate is determined by 2 nearest neighbors of each base pair that can mutate it. I denote the context with W. But then on top of this, independently, there is we add position mutability that can account for other factors and simply depends on absolute position of a base pair in the gene. And so since we have reconstructed the lineages and also phylogenies, our data is ultimately a collection of independent branches. And for each such branch, we can easily write likelihood, where we assume each site evolves independently. And so the likelihood of each branch of such phylogeny is simply a product over all positions in the sequence, where separately we write Poisson probabilities that a given position is constant, so nothing happened, or the complementary probability that a position has mutated. And here, again, the mutation rate is a product of the context-dependent times position-dependent mutabilities. And we can maximize this likelihood going over all branches in all lineages that we reconstruct from the data. And in the interest of time, I will only mention that the context-dependent rates are in agreement with previous models that actually looked for context dependence in productive data. So what was developed here was a more sophisticated procedure to avoid the bias of selection. But let me focus on position dependence, which is the thing that we added to our model. So this is the profile of these position-dependent mutabilities across the position, which is counted as the distance from the 5' end of the V gene. And what we see is that as long as this profile is clearly very heterogeneous, we see a significant enrichment of the mutation rate in CDR1 and 2, which are the 2 regions encoded in the V template that actually, along with CDR3, determine the binding properties of the receptor. So again, this is data that is free of selection, since these are out-of-frame lineages. And this would suggest that the mutation machinery is actually geared towards optimizing the sequence in the way that absolutely makes sense, because of course, what we'd like to modify are the regions of the sequence that code for affinity. And this effect of enrichment or enhancement of mutations in the CDR regions, we can see whether we define the position as a distance from the end of the V gene or the opposite end of the J gene. In both cases, the positions of the complementary determining regions 1 and 2 correlate with increased mutation rate. Furthermore, we used our model, which comprises context and position dependence, to study the phenomenon of co-localization of mutations. So again, we take our phylogenies, and now let us focus on a single branch again. Here we see 3 mutations that are marked in red, and we ask what are the statistics of the distances between these pairs. And so maybe first, the way we're going to measure this is with the correlation function, which is defined simply as the number of pairs that are distant by a given distance r base pairs, normalized by what our context and position dependent model predicts for this very quantity. And now if we look at pairs of mutations that are in different branches, so pairs between these red mutations and all the other green ones, we find this green profile, which is roughly equal 1, which means that our model does a good job at explaining the distances between different mutations. And on the contrary, if we only build the statistics out of pairs of mutations within branches, we see an enrichment at neighboring base pairs, which is of order 5, which is quite significant. Now, to get a sense of the scale of this effect of co-localization, we built a really minimal model where we simply say that each targeting event that follows the law described by the model I talked about just before can be followed up by a number of mutations at neighboring base pairs, where neighboring is defined with this exponential kernel characterized by width psi, and then overall probability to mutate epsilon. And we find that to fit, to match the profile we observe in the data, we can do so with parameters that correspond to the correlation length of 10 base pairs and probability of mutating 5%, which summed over all positions where follow-up mutations can happen gives us total probability of order 1. And this we interpret as a sign that, in fact, of a given number of mutations that we observe in any given lineage, this number of mutation is actually different from the number of targeting events that the sequence has experienced, or the sequences have experienced, and there is roughly 1 follow-up mutations per each targeting event. Finally, we revisited this phenomenon in productive lineages. So here you have 2 productive lineages from 2 individuals in orange and blue, and we see a further enrichment of nearby mutations in these 2 individuals as compared to the out-of-frame signal that I described before. This suggests that the phenomenon of co-localization is in fact useful for speeding up affinity maturation, as of course the neighboring base pairs and then the neighboring amino acid residues are in contact and in fact conspire together to build high affinity for a given target. And therefore, it absolutely makes sense to co-evolve them, to change them simultaneously. And hence this enrichment here is particularly visible at the multiples of the distance of 3 base pairs, which is of course the length of the codon. So finally, I'd like to move to a project that we've now started and is still an ongoing work, where we try to use our models of recombination and mutation to do a better job at identifying clonal families. So the standard way of doing this is typically to group the sequences in the repertoire sequencing data into VJ length classes, where length depends on the CDR3, where length refers to the CDR3 length, since of course, if we hypothesize 2 sequences are in fact related and stem from the same recombination event, they should share these 3 characteristics. And then within each such class, we study the Hamming distance between sequences, as is the standard way, and we identify this bimodal structure where the first peak depends on, is there because of the related sequences, and its width is due to the mutation rate, whereas the second one is simply distances between pairs of independent recombination events. Now, second thing you see in this plot is that this distribution is stratified across CDR3 lengths. And you can see that if we'd like to separate related versus unrelated sequences, so the ones that share the same recombination event versus the one that follow from two independent recombinations, choosing the threshold should probably be adapted, first of all, to the data set, and here there are existing methods to do that, but also to the CDR3 length, since as long as the mutation peak stays fairly in the same position, the one that comes from independent recombination seems to move along as we increase the CDR3 length. So we set out to study this using the model of recombination. And so we generate sequences from the model of recombination. And this way we can obtain very many pairs of unrelated sequences. And so here we have the second peak only, since none of the sequences are actually related. And the model allows us to estimate this distribution very accurately. And in particular, we can measure at which threshold of Hamming distance should we cluster the sequences together to achieve high specificity. And in particular, so the idea would be that if we cluster all sequences, join together sequences that are closer than 5 base pairs away, sorry, 5 mutations away, or 5 substitutions away in the CDR3, we would get partition to clonal families characterized by high specificity. This, of course, choosing such thresholds, of course, translates to a given sensitivity, because the closer we are to 0, the more conservative we are in our choice of threshold, the less of the signal that's present in the data we can recover. So what we did is we measured where this high specificity threshold should be across different CDR3 lengths. And that's the result of this, where here the red line is the high specificity threshold. So the one here that ensures the specificity is larger than 99.9%. And as we see, this threshold grows faster than linearly with L, and in fact, it's a quadratic dependence. On the contrary, under the hypothesis of roughly constant mutation rate, the threshold that ensures high sensitivity simply grows linearly. And so this allows us to see that there is a region or part of the data set, namely the one with long CDR3s, where high specificity threshold is actually above high sensitivity threshold, which simply means that any threshold I would choose in this yellow phase ensures both. And so in this sense, Hamming distance is enough to have a partition into families that's both high specificity and high sensitivity. And then on the contrary, there is this frustrated region here, the short CDR3s, where Hamming distance is not enough, because whatever threshold we choose, we either hurt the precision of our inference, which is to say we join unrelated sequences together, or if we adapt a high specificity threshold, we end up with very poor sensitivity. And so this is how the statistics of recombination allows us to view this problem of families inference. And here in particular, we know that we need to do better than Hamming distance in the CDR3. And this is possible since since what we measure, since what sequence is not only the CDR3, but also the templated regions. And in particular, we see how many mutations in the templated regions the two sequences have, and how many of the mutations are in fact shared. And so by writing down likelihoods, and I'm sorry, but I'm gonna essentially skip through that here, we can write the likelihood for the two hypotheses, the one that the two sequences are independent here, and the ones that they are related. And this allows us to build an expectation of, in this case, the number of shared mutations or the CDR3 Hamming distance given the levels of mutations. And what we end up with after this combinatorics exercise are particular and distinct scalings of the two quantities in terms of the mutation load. And so here is just a simple example where we test these ideas with synthetic data. And so these expectations that we have that stem from our models of mutation and recombination allow us to rescale these variables according to these natural expectations in terms of N1 and N2, the sequence-wise numbers of mutations, and find a separatrix between the positives and negatives that ultimately gives us much higher sensitivity at high specificity for fairly extremely short CDR3 lengths. So with this, I'd like to conclude. I think I've showed you that probabilistic models can be useful to describe, in particular, the process of somatic hypermutation. We found signatures of the mutation process that suggest that this is actually geared towards speeding up affinity maturation. And finally, that these ideas may also be useful to identify B-cell clonal families in the first place. Thanks a lot.
Thanks so much for the very nice talk. Are there questions from the audience?
I can ask a question. So indeed, a very nice talk. Thank you very much. Well, one major question is, do you have any hypothesis of what causes this apparent position-dependent mutability model? And the second part of the question is, well, why doesn't it appear also in CDR3?
Yes, so the one idea that we had is perhaps that what we pick up as position dependence could also be context dependence, but the one that doesn't fit in our 5-mer definition of it. So if we imagine the context dependence would be in reality 7-mer or 8-mer dependent, then this signal would in a sense not fit in our narrow definition, and then should CDR regions be increased and should have more hotspots, then that would translate into this upturn. And then, of course, it's known otherwise that it is indeed the case that CDR1 and CDR2 are enriching hotspots with respect to the framework regions. And then for CDR3, that's not the case because, of course, it's much more scrambled in terms of motif content because it's just simply much more diverse. So that could be one explanation, but I can also imagine that the mutation rate is simply modulated in terms of absolute position.
Yeah, the second answer I understand, but I don't understand the mechanism. How does AID knows, where the CDRs are? But
no, yeah, that's true. I agree. It's a bit.
I can suggest another explanation. I mean, what you basically said is that maybe the hotspots are not defined in 5 MERS, but rather in 7 MERS. But based on the second part of your work, I mean, the second part of the first part, you observed some kind of clusters of nearby mutations. So maybe these kind of clusters create a nonlinear effect. So once you have like a hotspot, it accumulates more mutations, and then it's not anymore a linear process, and then you can get some things like this.
Yes, so this, and in fact, we tested for that since we concluded that many of the mutations are in fact not direct product of targeting. And so we then created simulated data where we built, where we actually target only some of them and then build these follow-up mutations independent of context. but we actually don't see any change in the position dependence.
Okay, interesting. Thank you.
Any more questions from the audience? Then I have a question for the second part, was a clonal family inference. So I found it interesting that you mentioned that you wanted to improve on the previous methods that are around. So what is the evidence that those don't work, basically? Did you benchmark, or what do you see that is wrong in the previous?
Yes, so there is in fact quite a number of methods by now. They differ in terms of approaches they take, and there are in particular likelihood-based methods, importantly from the group of Eric Matson, and then other methods, several from the group of Stephen Kleinstein. I think what we'd like to do is to sort of find maybe a new compromise in terms of accuracy and also the speed of these methods. But so we have these ideas which are mostly concentrated about this around the difference that the CDR3 length makes. And we are currently trying to test, as you say, benchmark the scalings that we get with respect to this sometimes more sophisticated methods.
Very nice, very nice.
Yeah, just a comment about this. I think a paper from Steve Kleinstein's lab in 2015 maybe mentioned something similar, that you should do this kind of, they do it in a different way. They do it like this to nearest, and you're doing just the whole distribution of distances. But then they claim that the threshold is also a CDR3 length dependent. So I think it's similar ideas.
Yes, in fact, choosing the distance to the nearest neighbor is really something that requires careful fitting, because of course the extreme value statistics will tell us that this is going to change as we change the size of the data set. Yeah, absolutely.
Any more questions? Then I think we can conclude. We are also perfectly on time, which is great. So then I think we thank again our speakers, Kathy and Natanael, for their very beautiful talks. And I think this was the last seminar for this year, and then we will return next year in January again. Very exciting. Okay, thanks everyone.
Thank you. Happy holidays.
And there was a question about the recordings. There is now a new, I think, iReceptor YouTube channel where you can find them. We haven't, I think this has not been publicized broadly yet, but they are, yes, Pam is answering. Yeah, yeah. It's on the website of the iReceptor. Yeah, yeah. Yeah. And Gu, you wanted to say something? No.
Just happy holidays to everybody.
Happy holidays. Yes, happy holidays, everyone. Bye-bye. Bye-bye.
Bye.
See you.